This is a production of Cornell University.
So what I wanted to talk about, however, is and I know that I usually give talks about the research that I do, but I was invited by Neil to talk about teaching today because I'm teaching plant physiology at the horticulture department. And the plant physiology course, I will go a little bit into detail. There is a course that is more or less the only physiology course in the plant area for entirety of Purdue University. So everyone who is doing plant sciences there, and we have quite a number, like you guys here, I assume, quite a number of departments from forestry all the way to vegetable management and so forth. They have to take this course. Have to is a very important part of that. I have the honor to teach these students. And I will tell you a little bit about what we did in the last few years to try to make this course a little bit more attractive and more effective for the students as well as for the instructors actually. And then also what we did in terms of informal learning situations for our students as well as other students outside of Purdue University. So when I started out teaching at Purdue, it was like the typical career that each and every assistant professor has. You get thrown in the water is pretty cold, and you have to swim. So nobody really instructs you how to teach a course. I had the advantage that I taught quite some courses in Germany as well as in Arizona, so I had a little bit of a background in teaching everything that is considered green, so from plant anatomy all the way to functional genomics and plant. But I realized really quickly that there's always an instructional problem, or often an instructional problem that we are encountered with if it comes to teaching. There's usually a learning gap that the students have because some of them, and especially in this course at Purdue, some of them have very little background, very little knowledge that they bring with them either from high school or from other courses that they took in their departments, and others are already quite advanced. So how to challenge the students with a solid background knowledge and still don't let the ones who don't have this much of a knowledge behind. So what actually is the real problem here? And the real problem is certainly, as you can see, students. They're always the problem. It's never the instructor. This is what I learned here. And in this case, the diversity of the students certainly does not really help to have the course and teach the course in a very effective manner if you just go with the traditional methods of teaching, like a lecture, 3 lectures a week, and then a lab that is about 2 hours per week in classical plant physiology experiments. So we set out to kind of address this learning gap that we found, in some cases at least, and we tried to help the students how to learn. Because in the course that I'm teaching, we have students that come from a major that is called Horticulture, Landscape and Design. It's actually our more or less our bread and butter major in the department. And these students have a career path that usually leads them into the greenscaper industry. They maintain golf courses after their career at Purdue. So it's a major that is kind of close to vocational education. They have very little introduction courses in the non-biology and non-botany areas like chemistry, no lab experience, and usually also very little experience in botany courses besides the plant physiology. And then on the other hand, just one example, but I could also pick the biochemistry students that are in our course or biology students that are in our course, the horticulture science students also from our department, and they have already under their belt 8 courses in chemistry, biochemistry, genetics, and so forth, and at least 3 lab courses and 4 intro courses in different fields of botany, such as plant physiology, plant pathology, and soil sciences, and so on and so forth. So that simply illustrates a little bit the two outliers that we have here in this population. And we thought about teaching tools, and with teaching tools, we thought we should have something that is a common point of reference for all students in the class. And you can say, oh yeah, that's the textbook. Yeah, you know, I mean, for one, the students always complain that textbooks are expensive, and I agree with them. Some of them are actually quite expensive. And on the other hand, you cannot guarantee that the students have a very positive attitudes toward reading 300, 400, 500, 600, or 700 pages of textbook per semester. So we thought we have to find something else that can serve as common reference point. It should also allow a teamwork approach to solve scientific problems. Very often we realize that the students lack a lot of hands-on experience in terms of learning. I mean, my attitude is you learn very well in a lecture if you pay attention. You might learn even better in a book because you can go back and forth, and you learn the most if you work not with your brain but with your hands, because somehow they are better connected with your brain than anything else, I think, ears and eyes included. So the hands-on experience really gives the students tactile experiences and haptic experiences that really can translate into an experience that then also increases their scientific knowledge. It should integrate individual talents in a group, and this is something that to address is really not that easy, because it is learning styles. It means simply translated learning styles. So some students, they go into your lecture, they sit down and they get everything that you say, and they can memorize it, and they can work with that, and it's great. And others say, man, you know, your German accent, I have no clue what you're talking about. You are ugly, you just run around all the time, and it just makes me totally dizzy to even sit there in these lectures. So I'd rather sit at home, read my book, and get what I need to get. And others have different kinds of learning styles that then also help them to learn the topics that are in the course. So the teaching tools should also ease procedural questions. So very often, and this is also acknowledging the learning gap or the knowledge gap, very often some students know already how to use a pressure bomb, for example. We discussed this point a couple of minutes ago. Others don't even know what a pressure bomb is, and they think it has something to do with terrorism or invading other countries and so forth. And also, it should initiate content questions so that the students can beyond what they learn. It's not a regurgitating of knowledge that they get from textbooks, protocols, or whatever, but they should start thinking about what they do and work with that. And then also it should allow, and this is maybe going back to point one, a repeated viewing and learning without instruction, so that they are kind of self-motivated and self-activated in terms of the learning. So the objectives that we set out were the following. We said basic terminology related to lab and research procedures should be taught with our teaching tools. Single basic steps in lab and research procedures should be addressed. Simply they should know what they need to do when they want to do an experiment, for example. They should be able, if they use the tools, to carry out basic steps in lab and research. Most underrated and the most important point in any kind of teaching, that at least what I do, is that students get a positive attitude towards the course, and in my case, because I like to teach science courses, that they get a positive attitude towards the understanding of scientific discovery and the process of scientific discovery. How do scientists think, and why is it important that particular things need to be learned, and how do things fit together, and how did people get these ideas that then led to the photosynthetic electron transfer chain that I now have to learn and to study. And it's really hard because all these particular proteins in there, they have these funky names and they can hardly memorize them. So students and instructors need to be able to identify the efficacy of the teaching method. I mean, you can not just change something and make something new and it's kind of spiffy and it looks nice and you think, oh, that's awesome. They have to tell you that it's awesome, otherwise it doesn't make a whole lot of sense. It should generate a lively discourse about the learning success so that the students can think about, did you really get the point? Is it really better than we had before, or is it just a try that was an error? And also, the students should achieve a certain degree of ownership of the course and success. Going back to here, the attitude towards the course is certainly fueled and positively influenced by the degree of ownership that a student has. It's not only a course that he or she goes to, but it's their course, and they contribute to something to it, and they get something out of it, and they can measure that, and they can tell you about this. So this is here for the teaching researchers amongst you people. I assume most of you are from the horticulture department and not doing that much in this area. But if you talk to people who are interested in pedagogy and developing teaching tools and dealing with teaching every day, the most important and the most heard term that you will hear is assessment. So you have to create categories that allow you to simply quantify what the change in the student knowledge, attitude, and so on and so forth is. So the easiest way, or the simplest way to do that is certainly to create questionnaires, to hand them out to the students, to let them fill in, then to get them back, and then to think about, was that helpful that we asked these questions? Do these questions tell us something about it, about the effect of our learning tool and teaching tools, or do we have to change that? And what I show you here is only the first series of questions that we asked the students after we implemented a change in the teaching. So we asked for videos that we made, because we were creating digital videos. I will show you a couple of examples of that. And we integrated the videos in different steps and at different steps of the course. So I will give you an example of a instructional video that is teaching the students the content, or is, let's say, interesting the students of the content of one particular classical lab that they took and that they had to go through. Another video is a procedural video that explains to them a device. And in this case, it's a device that is not that easy to work with, so they should really know what's going on, because otherwise they might blow them up. They might blow themselves up. And the third was a video that is more a narrative that presents a project that a group of students have done in an independent research project that we integrated as part of the course. So. For example, and I will come back to that, to the assessment, the outcomes that we expected from we called them video podcasts, were that we wanted the increasing participation of the students in making these teaching tools. We thought that this would help to also give the students ownership of the course. So they are not only there to be exposed to your changes and to take what you create for them, but they are also involved in the entire process of producing, as well as implementing, as well as assessing the tools that we generated. In a lot of assessments, and when I teach that course, the Plant Physiology course, what I usually do is I use different tools to figure out if the students are satisfied with how it goes, and if they can address particular positives or particular negative points during the course. One is the 1-minute essay. It's very simple. You give a piece of paper and a pencil to all of the students at the beginning of the course. They have 1 minute to write a buzzword or a sentence down, what they think is positive in this course, and they have another minute to write a buzzword or a sentence down, what they think is negative and needs to be changed in this course. Then you simply collect them all. You go through all these kind of points that are thrown at you, and then the next in the next lecture, so 2 days later or so, I go back and I present them the result of this assessment, and also how I think I would be able to address their criticisms or their needs, their requests for changes. And one request that I got a lot was the traditional PowerPoint presentation has its connoisseurs and it has its critics. And very often you hear, oh yeah, PowerPoint is all prefabricated and just go through it, and is, I mean, I don't have to waste my time here sitting in class. I can also sit at home and look at the slides that you're uploading on the website. And this is certainly a valuable point, because some students we talked about, or I talked about learning styles before, will have that learning style and will be much more positive with a convenient learning style, having the PowerPoint presentation at home, just looking at it and then being fine with that. And therefore, we also try to assess, can we use another kind of tool, like a video, like digital videos, for example, as a replacement for a PowerPoint presentation that is going through the topic of a chapter or a week or whatever. The dissemination of the products, of the videos, to the scientific and the science education community as wide as possible. And this was actually quite interesting. We thought about, okay, so we made these videos, and we thought they were really cool, and a lot of people told us, oh wow, they're really cool. And so we thought, okay, so we need some feedback on that, and how can we make sure that these things will be seen by a lot of people and then discussed by a lot of people. So we started out to present this as educational projects at the ASPB meeting, and actually got invited to create an educational booth at one of the ASPB meetings, so that the colleagues in the field could see that and discuss that with us. And then we also put these videos that we produced on YouTube and on some other pages that are also distributing digital content. And I think I checked it like 2 weeks ago. In total, we had 3 videos that we uploaded. We got about 20,000 hits from all over the place. And get also responses in terms of emails or commenting points from all different kinds of people that use these materials in different ways, not only for formal education situation, like a college class, but also high school teachers or 4-H, FFA, and similar things here as well as internationally. I mean, I was quite surprised to meet somebody from New Zealand at the last ASPB meeting, and she kind of scooped me out and said, hey, I've seen your videos. I like that and that, I don't like that and that. And I said, oh wow, that's amazing. I mean, New Zealand, that's quite far, but for YouTube, it's just a millisecond more than coming from Purdue to Ithaca, I think. And then certainly to collect and analyze evaluations of the podcasts in the lab classroom and incorporate possible improvements into the teaching technology. So an ongoing process that allows us to comment on our own experience and also improve or add to what we have done there. So this is the first example. It's a narrative video. I should comment a little bit on this. So what we did after we realized that the students were really unhappy with these classical plant physiology labs, like you isolate chloroplasts and you show that they are doing photosynthesis, or isolate starch and show enzyme activity, and you have 2 hours time for that, which is what we had in our course. which is simply not enough to do something sensible. So you can only create demonstration experiments, which in my mind do not really do the job, at least not sufficiently. So we changed that, and we gave the students the opportunity to do independent research project where we simply scrapped the entire scheduling and said, okay, you sit together with one of the TAs or one of the instructors and then go through the entire project, create a hypothesis, create an experimental plan, make sure that you get all the materials, the literature, and so on and so forth. So it was a several weeks long process of preparing the experiment, and then we made sure that they had all the materials and facilities that they needed to do the experiment. And in the end, they had to present their findings in an oral presentation. We made a little bit a scientific conference for the course, and each and every group had to present their data, and each and every individual student had to write an individual protocol in terms of, in form of a scientific paper. I mean, with the structure of scientific papers and so forth. So what they learned was not only to kind of try to understand the scientific process of thinking, of hypothesis-driven research, but also how to organize, I mean, as a team with each other, which is sometimes pretty hard, as well as material, as well as being spiffy enough to get the facilities that they need, reading papers, discussing these with instructors and TAs and so forth, and in many cases going out of our department and talking to colleagues that had the microscope that they needed, or the photometer that they needed, and the postdocs that they needed to do particular things with them and help them to do their project. So what we all do in a normal situation when we have a research project where we need something that we cannot do ourselves. And so these three students here thought about an experiment that they were very interested in that was very applied, actually. They thought about making tofu. So they made tofu. The idea was not to make tofu. The idea was to try to find if they can show transgenes that are in transgenic soybeans that are in the tofu that they make. So they made tofu out of transgenic and non-transgenic soybeans. And then they went out and bought tofu from Walmart and the health food store and so on and so forth, and used a PCR-based method to detect the transgenes in all these food products. And they made a presentation of that they thought would help them to explain, particularly to their peers, but in addition to that, to their families and friends and outside people, outside of the classroom, what it is about what they do. Because they told me very often they get the question, so what are you doing in this plant physiology course? And they, oh, I do PCR and I show the glyphosate, the resistance gene, the Roundup Ready gene in soybean plants. Oh, okay. Yeah, just go and do that. That's a good thing, I think. And that's about it. But they wanted to have something that is a little bit more telling, and so forth. They created this video here. Let me see if I can start that appropriately. Just relax and take it all in. It's about 5.5 minutes or so. It's called Are My Soybeans Wearing Different Genes?
Are there weeds in my soybeans, or soybeans in my weeds? Alright, team, what have we got?
Well, the soybeans look exactly the same.
Although this one has weeds and this one doesn't. There's got to be something different between these two plants.
Let's take it back to the lab for an analysis.
Sounds good. You guys take the plants.
So the idea was to emulate the crime scene investigation. So it's a plant science investigation that they have there. And they also emulated the roles that they have in this
I
TV
don't see any
series.
difference here. Yeah, we're gonna have to look deeper, like into the genes.
The cute
What's
bond,
a gene?
the smart.
Gene is the first name of the former Purdue basketball coach.
Following material that's in DNA. No, something in DNA.
So these are computer science students that they interviewed for this video.
We have to get the DNA out of the plant through DNA extraction.
So you see now, this is not a one-to-one explanation how PCR works.
I'm extracting DNA.
Hey guys, come here. I think I've got it. We have transgenes.
What's a transgene? A transgene?
Again, computer science students.
I have no idea. Honestly.
I don't know what trace genes are.
And the animations that you see were also made by these students. So they produced the entire material that you see in this video.
So the resistance gene allows the fungus to then apply the pesticide over the crop without damaging the crop and still kill the weeds.
Steve, what are
You
we
haven't even
scientists?
used the Roundup gene in your tofu.
Pretty highly likely because 95% of the soybeans that are grown are genetically modified.
Wait, hold up. You mean I'm eating DNA and genes?
Probably every day. I made food, there's.
Or in fruits, food sciences.
We should test your tofu to see if it contains the Roundup gene.
Can you really do that?
This is where we can use PCR. It takes one copy of a gene and makes multiple copies.
I see it. I see the gene. Yeah, the gene's there, but you can only see it once you run it through a gel. I'm making a gel.
These bands are the Roundup gene.
Right. This first lane acts like a DNA ruler that helps us to measure up the size of our genes. This lane is empty because there's no Roundup gene in the plant. This last lane is from an organic tofu, which doesn't contain the gene either.
This band is from the soybean without weeds, and this one is from your tofu. You can see they both contain the Roundup gene.
Makes sense, huh?
So we solved the case. This soybean has the Roundup gene, and this one doesn't.
Great job, team. Another piece of mystery solved.
Okay, so this was this was the first example that I wanted to show you, the narrative. Important there is, or what I not important there, but what I also want to mention is, it is not only that, and I will go into the details in a couple of minutes. It's not only that they did the production, they did the acting, they came up with a script. They also made the animations that you have seen, and actually the design. And I will go for this also. The design of the marketing tools that they generated was also from them. But we also had a high school student actually who did the musical score for us for the video. So everything is handmade, and everything involved the work of the students from the very beginning all the way through the process to the last step. And let me show you that. Oops. Come on. Okay. Let me show you this in a couple of minutes. What I want to do here is go through a very brief discourse about a discussion that I have with the students very often in this class. So one question that always comes back, and specifically from the students who have ideas already about their career, and the career path is not scientific research, is why do I have to study plant physiology? I mean, that's good for nothing. It is complicated, and I get headaches from that. This is what I usually hear. I mean, I also heard I hate science and all that, but I don't want to mention that too much. But only a little bit. So what I try to do here is, and these are slides that I developed with the students, and I do this usually at a Blackboard session, that I simply throw at them, okay, you guys tell me the 10 most pressing problems in the world right now, and I will show you how we all solve them with horticulture. This is about the idea that we have here. Thank you, thank you. So what always comes up is global warming. And I mean, it is obvious that there's a lot of ways to think about CO2 production, to think about CO2 sequestering with plants, and so on and so forth, and how much plants have an effect on water evaporation, cloud formation, O2 generation, and so on and so on. It's a no-brainer in a way. The next point that is always made is certainly biofuels and renewable resources. And I mean, I think I don't have to go into details here. I mean, we all are very familiar with these themes. The next is human health, food and nutrition, and especially in the horticulture department, we had food science as part of our department until 15 years or so ago. This is also phytochemicals, cancer treatments, herbal medicine, functional food, obesity, antioxidants. These are buzzwords that they throw at me or I throw at them to address this particular point here. Science education, a very important point, which sometimes is forgotten, but life science education, we know about STEM education and so forth, lifelong learning. This is clearly something that can be done with horticulture and with plant science. And plants are interesting. It's not only animals that they have at home as pets in Indiana. Some of them come from rural settings, so they know that plants grow on the soil and that food comes from plants and so forth. So they have an idea how to deal with that. Interesting, the political aspects, poverty and peace. Certainly people who are employed in agriculture, people who are self-subsistence, and have vegetables for a healthy diet, usually have not that many reasons to become that angry and go for somebody else's resources. So this is also a very important point in terms of global politics. And then certainly a question that is always coming and that you saw also addressed in this one video, transgenic plants and foods, environmental health risks of transgenic plants, safe transgenic technology, release of transgenics, and so forth. This is certainly always a question that needs to be addressed, and also we as scientists have a very important role there. We need to really go out and be much more preemptive and really go out and tell people about that and discuss this with people. Recreation and sport. We have a wonderful setting here around Cornell, and it has a lot to do with plant science and horticulture. National forest parks, city greens, golf courses, all these are aspects of this question. Chemistry, if we want to develop new technologies and new materials, special oils, rubbers, lubricants, dyes, and stains, that certainly will be done with plants. And the environmental questions, just 1 point, heavy metal remediation, reforestation, and so forth, but there are many, many others also. Certainly plants play a very important role here. So you see there is a whole hit list of the top 10 that you can make, and that I usually like to make with the students, to also alert them about the importance of plant physiology and what they learn in this course. Let me give you a second example. This now is a procedural video. This was actually the first that was made, again by students. The title is Pigment, and it explains the methodology of an experiment that the students did in the course. It is also shorter than the first one, and it goes through the experiment that they do. So it's a pigment extraction and an analysis of pigments, of plant pigments. And again, this is produced and conceived by the students in the course for a 2-hour experiment. So they have a flow chart, harvesting flowers in the greenhouse.
Note the original flower color.
And you see, you've seen things like that in your own classes, because you might have a similar experiment that you do yourself. Extraction of pigments. You see the frame in frame, so they see the procedure itself. And they see the single steps so much more clear than in the first case. Sorry for that. So it's a very classical experiment, isolating the pigments, putting them on the TLC plate, running the chromatography, drying the plate. There's a second experiment that they did, simply pHing different, pHing the same pigment extract with different pH steps, and see the structural changes in the molecules, in the pigment molecules, that then lead to different coloration of the solutions. And then they read the whole thing in the spectrophotometer, did a spectrum. In the meantime, the TLC plate is running and separating the different pigments that they isolated from in this case, petunia flowers. And in some cases, we then, after the TLC plate was harvested, we also observed it under UV light and found additional molecules that do not show up in the visual light, but show up in UV. So it's not always there what you see, or there are things there that you don't see. And again, the production depended on the students in the course and involved their work. The third example even shorter than the second, so I don't want to bore you to death with that, is a more distinct explanation. It's an instructional video. We also did measurements, transpiration and water pressure measurements with a pressure bomb. And for you guys who work with pressure bombs already, they are not that easy to handle. And you really have to make sure that the students don't try to kill themselves with it, because you work with very high pressure, and it is sometimes not that easy. So step by step has to be clear, and we always start with the first aid kit so that they know that we mean it. This is an instruction that they also get from the TAs, but they have this always at home. They can just log on YouTube and watch it again and again. There are no words in it, besides the written words down here. One example where it really worked. Usually you have to do 3 or 4 trials before you have the handiness to really make it work. Okay, so these are the different categories of instructional tools that we develop, addressing different questions. So if you really want to instruct the students very detailed about one particular device, one particular piece of equipment, if you want to explain and show at least one methodology, or if you want to have a narrative that is giving background about an experiment that they did. So what you see is, it is not an instruction that comes from the textbook. So without the written instruction from a textbook, these videos would not be as useful, because you might not get each and every step, because some steps you cannot depict in 2.5 minutes with very fast cuts and so forth. But these videos are thought about a visual hook that kind of pulls the students in to think about it, to prepare for the experiment, for the course, and to be engaged and be attracted to what they are doing. So how to make videos? One is that the students are engaged in each and every step. I said this before. This here, for example, is a design that I talked about for the plant science investigation, and the students also developed that design, put it on their lab coats and on front of their lab coats, so they were professional plant science investigators hired by the FBI or whatever, I don't know. So they also understood that they have to create a corporate identity to market their products, because they wanted this product to be out there and be seen. So what we then also did is we included these videos into competitions for scientific instructional videos, and they won the first prize for that. They were certainly very enticed by that. One of them actually started studying communication, scientific communication, and she is now on the track to become a science reporter or a journalist, a scientific journalist. I think this is actually quite an achievement from these setup here. So the video production, as you see, is quite a complicated process that involves many different steps, from the hardware and securing the hardware, to doing the soft part, the storyboard, to do the shooting with all its different aspects, and then the post-production. This is normally the 4 steps that you need to address. Let me go through some of them. So the first steps, you certainly have to have some video equipment. Nowadays, the video equipment gets cheaper by the day, so it's not that hard anymore. So you can even use a very good iPhone with a very nice video camera and simply shoot material that you want to use. A lab setting is helpful, and this here is essential. So we had 2 weekends that we met and really developed a script for the video. So we thought about, okay, what do we want to say? Who are the target audience? How do we say this? How long can it be? How short does it have to be? Is it understandable? How can we test this? Do we have a test audience, and so forth? And so we really went through it. We really went through it in a way of a professional filmmaking process on a very, 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 very small scale. The second step would be then the shooting, and you see here, I mean, decoration, prop, mask, and all different kinds of things, and then the post-production, the editing of the video material. I have to admit, we had a video artist that we enticed to help us with that, because certainly as scientists, you're not really usually very familiar with editing and these kind of things. And also, the musical scores that you heard in these videos, the students thought that these are really important, because they not only make it a little bit more entertaining and not that dry and maybe with a voiceover or something from a professor, but that gives you a particular rhythm. And when I stood there and showed you the videos, I saw a couple of feet doing that. And this is exactly what the idea is, so that you kind of get it into a automatic or with an automatic rhythm that is helping you to kind of not be lost after a minute or two, because the material is too complicated that you see in these videos. So then the third steps would be to disseminate the video, and I told you we put it on the web. And then also we used these videos as well as experiments that we did for outreach purposes. So we had high school and middle school classes that come in through the summer. We have the so-called HASA, Hoosier Academy of Science and Agriculture, as well as a FFA and 4-H, high schoolers and middle schoolers that come in and do courses. And in these courses, we use the video material as well as simple experiments that they do, because, as I said, I think that working with your hands, you learn much, much more than just working with your brain. Another point, apart from the videos, and only connected to it because you saw one of these experiments depicted in the first video, my soybeans wearing different genes, is the aspect, and I want to mention this only very briefly, the independent research projects in plant physiology. So I told you that using the classical experiments in plant physiology, going through all different aspects of plant life, if you want, at least the molecular plant life, is certainly not very attractive and sometimes tedious, especially if you have a lot of time constraints, like only 2 hours per topic per week. So we decided, as I told you, to break this up and let the students do their own independent research projects that are planned together with a TA or with an instructor. And the way I thought it would be very effective is if we have grad students and postdocs who kind of deliver a project out of their active research programs. This has there are 2 different aspects that come with that. For one, you have a person who is really invested into the project, because that is certainly something. Maybe it's a question that the grad student wants to address. Maybe it's an experiment that he or she has to reproduce to get the paper published or something like this. And also, you have the person with mostly the highest expertise that you can find in this particular area, because the grad students very often know more than their advisors, at least if it comes to the practical parts of experiments. And also, the advantage in these cases is that you have a peer-to-peer situation that is much more peer-to-peer as if it would be the professor talking to the undergrad. So having somebody who went through the same kind of experience just recently, a year or 2 years before, can certainly relate to the learning and needs and to the particular requests that the students have in totally different ways than somebody who has been in the lab maybe not for the last 15 years or so. And so this is kind of a big brother, big sister approach, if you want. So you have somebody not that far advanced in the career that is able to relate and to talk with the same language. And this is something that I want to emphasize also. It is very important when you create these digital videos, for example, or other digital material, that the visual language has to be the one of the students, so that they understand. When I saw the pigment video for the first time, it was even cut at a faster pace. I told them, I have no clue what you guys did. I will never get anything by watching this video. And they said, oh, no, maybe it's a little bit too fast. We'll make it a little slower, but that's the right pace. I mean, it has to come with like flashes of a couple of seconds. So they are used to watching YouTube and so forth videos that are all cut in these particular ways. The visual language that somebody who grew up with a radio or a black and white TV and the internet is certainly totally different. And so that needs to be addressed. And also the ability to have music and a score in the background that kind of hammers in the content is also important here. So what we try to achieve here is to again invest the students in what they do. So they have really something to decide what they want to do, how they want to do it, who is doing it, and so forth. And what you see here is actually an experiment that is on quite a scale. This is 70,000 EMS lines of maize tested in dark grown experiments on a response to a growth inhibitor. And these were 8 students who worked through 1 semester on this. This is actually here seed bags of the lines that you see rolled up here on a piece of rope. And you see these flats and flats and flats of material that then had to be taken out of the dark and photographed and then go through the analysis. And they actually found a couple of mutants there that were quite helpful and presented the data on different occasions. So I want to thank specifically Ellen, Carla, and Emily, who were really driving forces in the entire video project. Diana Nacera was the video artist that worked with us, and Stephen worked on the first set of videos. If you want to learn more about this, you will find a website at the Horticulture department. And I can also, if you want, give you details on that. And you can certainly always go on YouTube. All you have to do is you have to type into the search line Schulz, and then if you, yeah, that's very romantic, huh? and then you click and you find all 3 of these videos for you to watch again and again and again. I want to make one more remark before I finish, and then I really do finish. The shoes that you see, that is also on purpose. So you will maybe forget what I said, and you will forget where I came from and who I am and so forth. But you will think, man, you know the guy with the funky shoes? He talked about these videos? I remember these shoes. Thank you very much. Go ahead.
My name is Burkhard, I'm also from Cancer. And first of all, I want to say that as a postdoc right now, I'm kind of in between, and I still have my experience very fresh as a student, and then still thinking about the future. So I'm kind of in between, and I have to say I'm very, very impressed
That's a great question. And I will address it in two different ways. First of all, no, it was not supported, at least not at the start. After we won the first award and we showed that this was really a very valuable way, I went to the university and asked them for funding. And they actually gave me some funding for video equipment and for some of the materials that we needed to do the videos. I would love to have the ability to hire a video editor for each and every of the videos that we are making. So that is something that is not possible with the funding that I got. But most universities, and I bet Cornell is very similar in this way, has a department of video technology, computer design, or communication, and so on and so forth. And very often the students in these departments, they're looking for projects that they need to do for their masters or whatever. And so what I'm doing now is I'm always reaching out to them and trying to engage them, at least for the technical part. Because certainly they, as you've seen with the computer science students, they have no clue what a gene is, and absolutely no clue what a transgene is. But they know how to make an animation. I mean, Carla made the animations here, but we already thought about and talked about with them how to improve that. I proposed this project on a different scale and a different form also to NSF, and it was funded as part of a career grant, so it will be ongoing through the next couple of years. And what I can say, what my experience is, because I was interested in this from kind of early on about teaching and also doing research about teaching. There are always institutions like Center of Instructional Excellence. I bet you have something here that sounds the same, and things like that. If you go there as many times as possible and try to participate and to benefit from the offers that they have there, in many cases that is quite helpful, and you get always new ideas if you talk with researchers of teaching or with instructors, teachers, educators, pedagogists, and so forth. And I now work with ASPB for undergraduate education on one of these committees that are involved in undergraduate education at the American Society of Plant Biologists. So you kind of, if you're interested in that, there are outlets that you can find where you find yourself and your activities addressed.
Which of these three video types that you showed, which do you regard as the most successful, and of course, what are the criteria for?
It's really funny.
Yeah.
I would say, so what do you guys think? Is it the soybeans, the pigment, or the pressure bomb? Who's for soybeans?
What's your metric for success?
Let's say how many people watched it, or
How about how much plant physiology they learned?
exactly how much plant physiology they learn. For example, how much the students are satisfied with what they did in these videos. So I would also say, yeah, the soybean video is very appealing. It is very entertaining and so forth. But actually, the one that resonated the best with the students, as well as with other people besides the students, is the pressure bomb video, because it is to the point, it is clear, and it is no bells and whistles. It really explains a procedure that they have to go through and they really need to address and to master. And I think that is clearly a way that you have to distinguish. So in your course, or for a different purpose outside of your course, for informal learning situations like engaging middle school and high school students, the soybean video would certainly engage them, attract them maybe, and kind of give them the idea that science and plant science is something that is really worthwhile.
This has been a production of Cornell University, on the web at cornell.edu.